This stuff is so crazy. Last year, I had the pleasure of joining a delegation to go to the Valley, and we spent a day at Singularity University, which is for me like one of the mind-blowing places. And they were talking about printing tissue, and it was just one of these things I just could not imagine. I mean, I understood the concept, but up to like 4 minutes ago, I've never seen the result. This is the result. That is absolutely bloody crazy, to be straightforward. It's actually an ear in there, like a printed ear that was printed in Austria. I can think of so many bad jokes related to Formula One, which I'll not make now, but we have an ear available. I don't know which side it is. So Nikki, if you're listening, anyhow, that was awful. We're talking about printing 3D living tissue. This is absolutely, it is crazy. Boys and girls, the CEO of a company called Biobots. Please welcome on stage, Danny Cabrera. Welcome on stage, Danny. And you know what you need to do.
Thank you.
My friend,
Thank you.
the stage is yours.
Imagine a world where curing a disease is as easy as typing a few lines of code, or where creating a brand new organism is as simple as using standardized software that runs right on your laptop, and all you need to do is press print. Doesn't that sound like the type of interface we're used to in the 21st century? Well, that's not the case for biology today. But if it were the case, and if we could do that, then you could imagine doing things like designing brand new drugs and therapies that could obliterate disease. Or even beyond that, doing things like engineering and designing organs out of a patient's own cells that could eliminate the organ waiting list. But why stop there? I mean, if we could really design and engineer living things, we could build organisms that can help us colonize other planets and really push life way beyond Earth. So why hasn't this stuff why isn't this real yet? It turns out that there are a lot of people working on these problems. Up on the screen, you can see Dr. Tony Atala. He's one of our scientific advisors, and he runs a lab at Wake Forest where they routinely create, engineer, and using a mixture of technologies, new tissues and organs that have been implanted into patients, literally saving lives. But the progress really has been fairly slow, and there are only a few of these groups around the world that have access to this type of technology. And there are two main problems. The first is that biology over the past 100 years or so has been done on flat, two-dimensional petri dishes like this. And now, you can imagine the body is this complex, three-dimensional living thing, and cells inside of the body are used to interacting in this space. And they end up behaving really, really differently inside of the body than on those flat 2D petri dishes. So that's problem number one. The models that we're using simply aren't good mimics of what's actually happening inside the body. And the second problem is that biology today is done by specially trained technicians. Basically graduate students and postdocs who spend years of their lives learning how to use pipettes and move really little tiny volumes of fluid in and out of plastic tubes. And now this isn't just expensive, extremely time-consuming, but it ends up being irreproducible. You can imagine, the rest of our manufacturing really happens, almost all of it, using robots. And biology is the only place where that really hasn't changed. Biology is still being done by hand today. If we really want to begin pushing forward and achieving some of these milestones and some of these goals, we're going to need to begin to blend robotics, computing, and biology, and really rethink the modern laboratory. Now, this isn't a new concept. It turns out that synthetic biologists have been, oftentimes compare biology to integrated computer circuits, only instead of zeros and ones, you're playing with DNA, RNA, and proteins, or the stuff of life. So I started Biobots with a friend of mine about 1.5 years ago. And our goal was really to begin doing this, begin building robots that could help digitize biology, or create a digital interface for biology, and also begin to empower people to do it in 3D. And up on the screen is our first product. It was a beta unit. As you can see, it was mostly 3D printed parts. And I'll tell you how it works. It works like a traditional 3D printer, in that it moves in 3 axes, X, Y, and Z. And it uses pressure to extrude material out of a nozzle. Really, that's how you are able to create your 3D living tissues. And the key part of this first device was that we used a new process for actually curing these tissues. You can imagine if you've ever seen traditional 3D printers, they use things like plastic. They print in plastic, and so they end up using temperature to melt the plastic. And this simply doesn't work when you have living cells because you'll burst them and kill them all. And the other method that's commonly used in 3D printing is using UV radiation. So I mean, you can imagine that's not very good either. I mean, when you walk out into the sun, what's the first thing you reach for? Some sunscreen, right? Because you don't want to mutate all your cells. So we developed a new process that uses visible light to begin to turn these liquids, turn these materials from liquids to solids without killing all of the cells. It turned out, it ended up being a really amazing device, and we had so much demand for it. We ended up creating this great beta program where we partnered with institutions, offered them the devices at a low cost, to really start seeding the world with them, and really made them start telling us what they were doing. That's really what we cared about. We wanted to learn about what the applications were, what was possible with it, and how could we work with these people to really understand how to make better and better products. So these are some of those early beta clients. We had incredible groups working on a really wide array of different tissues. I actually just visited one of our clients at Karolinska who's working on neural tissues, trying to recreate parts of a brain. I mean, and all of these, as you can see, are research institutions. So this is still definitely a research tool. And some of the type of work that people were able to do with it was do things like this: print really complex 3D geometries that were seeded with cells. This is actually using a biocompatible material. It's a collagen-like material. And if you were to touch it, it would feel kind of like your ear. People were being able to experiment with different material properties, really begin to understand how do we begin to mimic the tensile properties of the body. You can imagine the tensile properties of your ear are pretty different than that of a bone or a piece of skin. And really begin to examine how cells behave in 3D space. How do they live, and how do we begin to understand the systems and model them? So I mean, it ended up being a really successful program. We learned a lot from those early clients or those early partners. And off of that success, we were able to raise some money from venture and start growing out our team. And we grew now to a team of 9 people, a mixture of roboticists, software engineers, computer scientists, material scientists, and tissue engineers. And we started incorporating all of that feedback into a brand new device. And now the new device is the Biobot 1. That's what you see here up on stage. And it has a few key features that separate it from anything that we had ever done before. The first is really, as you can tell, it has multiple extruders. So you can suddenly begin to do multiple materials and begin to study emergent properties of materials as you mix them. It had temperature control, which allowed you to print different thermoplastics, which ended up being a really successful application. This is actually a piece of knee meniscus that's been printed using a thermoplastic and different proteins. And actually, when you implant that into a sheep, the sheep cells end up colonizing the cartilage and begin depositing their own material. And over a period of 6 months, the entire piece of plastic dissolves away, and you're left with a piece of knee meniscus that was actually built by the animal's body. And now this stuff is still in research, so it's not going into humans yet, but you can imagine the implications here. The other thing that we did was we kept the photo cross-linking module. So this is our bread and butter before. This is how you turn materials that are liquid into solid without killing the cells. And it's turned out to be an amazing way to create different tissues. And now we've begun really looking at next steps or next things that we can do. We've begun developing a catalog of different materials. We have now about a dozen materials that begin to be more standardized. And we're going in a direction similar to your inkjet printer back at home, where you have different colors, different colored cartridges. We'd like to be able to have someday different cartridges for different tissue types. And you can only imagine how much easier that would make things for people. And these are some of the types of tissues that you can build with the device. You can see the different colors represent different cell types from different tissues. And you can begin to layer them and control the geometries at a really precise scale. So this device is precise down to 10 microns, and those lines are about 100 microns thick. So now I want to quickly walk you through how you would actually use this thing and how easy it is compared to some of the existing technology that was out there before this. When you open your Biobot, you get a little box with a starter kit of material, which you mix up with your cells, and then you load it up into the Biobot. use any design software that you're comfortable with, be that open source software, Autodesk, SolidWorks, it doesn't really matter. And then you design your 3D geometry that way. Then you go ahead and import it into our software, which will convert it from a 3D geometry into printer instructions that the Biobot actually understands. And then you just press print, and the device does the rest. And up here, I have with me a sample that we actually printed yesterday downstairs. We were running demos in case any of you guys caught it. And it's an ear. It's actually Van Gogh's ear. One of our engineers back in Philly designed it, and we printed it out over here in Vienna. So I want to just talk briefly about this last piece that we've been working on. It's called Build with Life, and it's our community. So when we started out the company, it was really a group effort from different universities and different scientists. And we wanted to have an alternative method of really creating community and helping people get caught up to speed on how to do bioprinting and how to actually do biofabrication. So we ended up open sourcing most of our experiments that we run internally, and we keep logs of all those experiments right on our website so anybody can access them, learn from them, and contribute to them. So I know we're still pretty far away from a future where you can literally design and engineer any living thing on your laptop and print it on your desktop. But if that's something that's interesting to you, or you'd like to learn more, please come and find us afterwards. We'd love to continue the conversation. And with that, I'd like to open up the floor for questions. Thank you.
Now, let's see if we have any questions. Dear Slido, again, I find this so utterly mad. I mean, I asked earlier, how long does it take to print that?
So that took about 2 hours. I think actually Red Bull took a time lapse of it, so it'll be available at some point.
And I couldn't quite hear what you were saying backstage. Did you tell us why it's green?
Sorry?
Why is it green?
So it's green mainly for show purposes. So we used 2 different materials, one that was yellow and one that was blue.
Yeah.
So you can see like the emergent third property of color. We chose, the material is actually clear, and so you wouldn't be able to see it. So we added some food coloring in it so you could actually see it as we hold it up.
That's so crazy. Seriously, if you meet random people in a bar and they say to you, well, what do you do? What do you tell them?
I mean, I was doing that all of last night.
So what do you do? I mean, but not in a pioneer setting where someone says, yeah, I build space shuttles, yeah, I print ears. But like in a normal world, not in, I'm saying you're not normal, but like in a normal setting?
We build robots, right? So these are robots that are empowering people to design and engineer living things. And I usually just start with, we build robots, and if they want to know more, then I drop the bomb. You know?
And what's the reaction? Is it like usually, holy shit?
It's a mixture of holy shit and what the fuck is that?
But I mean, just what do you say then? I mean,
I can get into it. If they really want to know, I let it rip.
I mean, I'm just out of interest. How long are we talking until that'll be used on a daily basis?
Well, so the device right now is really a research tool, and people are really making or figuring out the foundations of how this technology works and how biology works. What's important is having the software tools to really capture all of that data and empower people to make intelligent decisions with it. So that's the stage we're at. It's still sort of the tissues that people are fabricating are much smaller than ears. They're on the orders of a few millimeters, except with the exception of that cartilage piece that I showed earlier. But that's when you're printing that, there's no actual cells that you're printing. The cells come afterwards, and they come from the body of the animal.
Crazy.
So as far as how far or how long is it going to take to have fully functioning organs coming out of bioprinters, I don't know what the number is, but we're still far away. Like I said, we're still in the order of magnitude of a few millimeters. And organs are a few orders of magnitude larger than that.
Crazy. My friend, we have a couple of questions here.
Right.
In what way or to what extent are the printed objects alive?
So, the input into the device, I don't know if I made that clear, but they're mixtures of biocompatible materials, things like collagen or gelatin, and living cells. So the cells are sourced either from patients or from animals or from established cell cultures in the lab. And so, when you start, the thing is alive. You have a tube full of living cells. And when you're done, you have those same living cells positioned in a 3D geometry with some material that you designed. So that's really the first step. After that, the tissue has to be incubated either in an incubator or in a bioreactor, and the cells then continue to grow and develop. And then it's only after that process, and that can take anywhere from a week up to a month, that you have a fully functioning tissue, or a tissue that's more mature. So the things are fully alive.
I mean, we've basically, we're so much behind time, but I'd just like to ask these two questions really super quick. Just super quick. Number one, are stem cells needed? Number two, how much does the whole thing cost?
Yeah, so stem cells are, depending on what your applications are, stem cells may or may not be needed. With the example of the knee meniscus, the answer is no, but it really depends on what you're doing. A lot of people do print with stem cells because it's a hot area of research, just trying to understand how do these cells differentiate. And it turns out that 3D geometry has a huge influence and a huge impact on this, as well as the stiffness of the materials and the cell and the other cells that are surrounding those cells. And then the second question is, how much does the thing cost? It's actually, the device itself is $10,000 and comes with everything you need to get started. It comes with software, the device itself, and a starter kit of materials. And the printing materials really vary in cost. We have a catalog, like I said, of about a dozen different materials. And you can go online and find the prices, but they're usually in the order of magnitude of a few $100.
Awesome. Danny, it's been mega. Thanks a million, mate.
Awesome. Thank
All
you.
the best. Thank you very much. Wow. Biobots. I am utterly intrigued by this. I think that's a lot more exciting than your random app, isn't it? No offense. I come from the app world, so I'm allowed to say that.